Hi, and welcome back. In case you missed my previous presentation, I am Dr. Hannah Lindsay of the Traumatic Brain Injury and Concussion Center at the University of Utah, and my expertise is in diffusion MRI post-processing and analysis. This is the final lecture of a three-part series on diffusion MRI for ENIGMA-U and should follow my previous presentation on white matter anatomy and the diffusion tensor. So I'm now going to go over several advancements in the field of diffusion MRI acquisition and data reconstruction, which have been developed to overcome many of the major limitations of DTI that I previously discussed. I'll then go through several of the most common diffusion MRI analytical approaches and provide you with some information about different tools that are publicly available for you to perform those analyses. One of the first big steps taken to advance diffusion-weighted imaging was the introduction of high angular resolution diffusion imaging, or HARDI. This is actually a data acquisition technique, and it just samples a greater number of diffusion directions than standard DTI, which typically samples between 6 and 32 directions and estimates a single tensor per voxel using a relatively low B value. In contrast, HARDI typically samples between 32 and 256 diffusion directions with a single high B value. Data is acquired across a spherical shell in Q space, which is a mathematical representation of 3D voxel space that describes the relationship between the diffusion-weighted MR signal and the displacement of water molecules, capturing both the magnitude and diffusion of, or direction of diffusion in a voxel. The higher angular resolution gathered using HARDI allows us to estimate something we call the orientation distribution function, or ODF, and that estimates the probability of water diffusing in multiple directions. As a result, HARDI resolves crossing fibers and provides a more detailed representation of white matter microstructure than DTI. However, because HARDI samples data using only one high B value, it can't distinguish between different diffusion environments, like free water versus restricted diffusion. So multi-shell HARDI acquisitions were developed, and these sample at multiple B values across multiple spherical shells. And by doing so, we can capture diffusion behavior at different scales, providing more biophysically meaningful metrics and disentangling contributions from free water, crossing fibers, and other microstructural features. Here you can really see the implications of HARDI acquisition relative to DTI. In the top images here, we're zoomed in on an area of the brain where you can see ventricles with a lot of free isotropic diffusion, like here. You can also see the corpus callosum, which primarily has one fiber direction. And then there's this area over here, which is called the corona radiata, and it has a lot of crossing fibers. So each voxel contains multiple fiber directions. The diffusion tensor does a good job in the ventricles with isotropic diffusion and in the corpus callosum, but over here, it doesn't really capture much information, and there's even places where it thinks it's isotropic and where in fact there's a lot of crossing fiber data there. Each voxel from HARDI, on the other hand, shows us the ODF in each voxel and gives us more information about those multiple fiber configurations, which you can really see the benefit of here. So compared to this isotropic value we're getting from DTI, we're getting these rich multidimensional ODFs. And down here are reproductions of the corona radiata resulting from DTI versus HARDI. And as you can see in DTI, we're only getting this primary trajectory of the corona radiata that just goes kind of up and down. And then with HARDI, we get a much more complete representation of the anatomy with all of the lateral projections and crossing fiber data included. The development of HARDI was really such a big deal because it allowed us to implement a large number of advanced models for reconstructing the data that we collected with the HARDI acquisition. These reconstruction methods generally fall into one of two camps. There's model-based and model-free methods. Model-based approaches rely on predefined mathematical models to describe diffusion signals based on specific assumptions about tissue microstructure. They provide interpretable biophysical parameters related to tissue composition or fiber orientation, pathology. And by modeling water behaviors like Gaussian diffusion, restricted diffusion, or multi-compartmental structures, they allow us to estimate tissue properties and perform hypothesis-driven analyses. They're particularly effective in simpler diffusion environments, but the reliance on assumptions can oversimplify the complexity of the underlying microstructure and limit their accuracy in more heterogeneous tissues. So diffusion kurtosis imaging extends the conventional diffusion tensor model by including additional metrics that describe deviations from Gaussian diffusion, including the apparent kurtosis coefficient, which represents the whole non-Gaussian diffusion component, and quantifies the extent of restricted diffusion within each voxel. There's also scalar metrics, mean, axial, and radial kurtosis. It's used to quantify microstructural complexity and heterogeneity in tissues, which makes DKI particularly useful in detecting subtle changes in pathology. It's also simple to implement and can be used with many existing data sets that relied on DTI, but it is limited by its assumption of uniform kurtosis within the voxels. Neurite orientation, dispersion, and density imaging, or NODI, is a multi-compartment model that models brain tissue as a combination of intracellular, extracellular, and free water compartments, which offers insights into microstructural integrity and organization. Its primary purpose is to provide biologically interpretable measures of tissue microstructure, and though it's highly informative, it does require longer acquisition times and is pretty sensitive to noise. The ball and stick model is another multi-compartmental model where water diffusion is described as a combination of one isotropic and several anisotropic compartments, which are represented as balls and sticks respectively, and it estimates fiber orientations using Bayesian inference. It produces metrics, including restricted diffusion imaging, or RDI, and the compartment volume fraction, which relates to the density of fibers within a voxel. It was specifically developed to improve fiber tracking by better representing complex fiber configurations, and it does this pretty well, although it is computationally intensive, and due to its assumption of discrete fiber populations, it might not really account for all the tissue complexities. Constrained spherical deconvolution is an advanced technique that deconvolves or removes noise from the diffusion signal to estimate the fiber ODF, or FODF, and that reflects the relative density or proportion of fibers oriented along different directions within a voxel. From the FODF, an estimate of apparent fiber density can be extracted, and this reflects the relative contribution of each fiber population to the overall signal. CSD is primarily used for tractography, and right now it's probably the best method we have for resolving multiple fiber populations and regions of crossing fibers. Its high sensitivity to fiber orientation and ability to reduce partial volume effects make it really useful for investigating structural connectivity, but its accuracy does depend on data quality and successful deconvolution, which means heavy computational demands and expertise. Additionally, it assumes some things about diffusion behavior which limit its applicability in pathological tissue. Another approach to the crossing fiber problem is to take a step back and characterize diffusion as a process itself rather than the fiber configuration directly. And various model-free techniques have been developed for this purpose. Model-free approaches use data-driven techniques to characterize the diffusion signal instead of relying on the predefined mathematical models. And they're particularly useful for capturing complex diffusion patterns like crossing fibers without imposing assumptions about the underlying tissue structure like the model-based approaches do. Model-free methods are all in some way based on a theory of Q-space, which describes the relationship between the diffusion-weighted signal and something called the spin displacement probability density function, or PDF. And that just basically describes the probability of water molecules displacing by a specific distance in a specific direction. To estimate the PDF along just one dimension in Q space, we have to acquire multiple diffusion-weighted images, each with their own distinct B value and diffusion direction. And so to fully characterize the PDF in all three dimensions of Q space, hundreds of distinct B values and directions are required. And this is super, super computationally intense. Nonetheless, the full characterization of the PDF in Q space is essentially what diffusion spectrum imaging aims to achieve. Specifically, DSI reconstructs the PDF by densely sampling diffusion signals across a 3D Cartesian grid. And this is the only reconstruction method I'll talk about that requires a specialized acquisition like this. The rest are pretty much reconstructed with single or multi-shell Hardy. DSI's primary purpose is to provide highly detailed and precise diffuse information. And this makes it ideal for resolving complex fiber geometries and for structural connectivity analysis. We can quantify various diffusion measures from it that measure restricted diffusion, such as return to the origin probability and zero displacement probability. We also get mean squared displacement, which measures the overall extent of diffusion over time and space, and generalized FA, which quantifies anisotropic diffusion in any direction and provides a scalar measure of the angular variation of diffusion without assuming a specific model like tensor-derived FA does. Although DSI produces exquisite diffusion data, the extensive and demanding acquisition protocol really limits its feasibility for use in clinical populations. Cue-ball imaging is an alternative model-free method that improves upon DSI by simplifying data acquisition and reconstruction and making it more practical for routine use. It relies on a single shell spherical Hardy sampling scheme, and it's 2 to 3 times faster and much less computationally demanding than DSI. Also, instead of the PDF, QBI estimates the diffusion ODF or DODF, which captures the angular structure of diffusion and indicates the relative likelihood of water diffusing in different directions. This allows for the resolution of complex fiber configurations, and we can still quantify metrics like generalized FA. However, because it samples on a single shell, it can't provide any measures of diffusion displacement or restricted diffusion. So even though it's faster and more practical than DSI, it's not nearly as precise. And also, its angular resolution is completely dependent on using a high B value, which can result in noisy data. Generalized Q sampling imaging, or GQI, was developed to bridge the gap between DSI and QBI by utilizing full Q space data through single or multi-shell spherical sampling, providing more accurate, flexible, and simplified estimation of the diffusion patterns while preserving details about fiber orientation and diffusion heterogeneity. GQI directly computes the spin distribution function, or SDF, which represents the absolute density of water molecules diffusing in different directions. And this simplifies water displacement representation while preserving angular information, enhances resolution, reduces artifacts, and improves sensitivity to complex fiber configurations because it's so computationally efficient. The SDF captures free, restricted, and anisotropic diffusion contributions within a voxel, and quantitative metrics can be extracted for each, including quantitative anisotropy, or QA, restricted diffusion imaging, RDI, and ISO, which is the isotropic component. And each of these reflect the density of anisotropic, restricted, and isotropic diffusion, respectively. Given that it has a multi-compartmental nature, GQI-derived metrics and the fact that it has, given the multi-compartmenttal nature of the GQI derived metrics and its ability to leverage multi-shell or full Q-space data, GQI gives us more biophysically meaningful and detailed insights into diffusion behavior and tissue microstructure, and this makes it so much more robust and versatile than QBI. Main advantage of GQI includes its flexibility in acquisition protocols and the balance it strikes between resolution and data requirements. However, just like QBI, it does sacrifice some of the detailed information provided by DSI to achieve broader applicability. So here's a summary table of the various reconstruction methods I just covered, and you can compare and contrast different features of each with DTI or with each other. Feel free to review it for a moment, take a screenshot, you can reference later. It also shows all of the DTI limitations that are addressed by each method. Okay, so there are several ways that we can analyze diffusion MRI data, and I'm going to briefly discuss some of the most common ones, which are listed here. They can broadly be classified into 3 categories: whole brain, voxel-wise, or region or tract-based approaches, but several methods do bridge these categories depending on how they're implemented, and there's no universally optimal method. Rather, the methods can be complementary to each other, and the most suitable approach is determined based on whatever your research goals are. Histogram analysis is an automated whole brain method that does not require any predefined ROIs or regions of interest, and it's ideal for assessing global effects on the brain tissue, which makes it useful in disease progression studies or evaluating treatment effects. It extracts diffusion metrics from all white matter voxels and then plots them on a histogram and summarizes the distribution. You can extract parameters like mean, median, peak height, skewness. All of these can be extracted and compared across groups using inferential statistics. And while it's efficient and provides a concise overview of tissue microstructure, it does lack spatial specificity, obviously, because it's whole brain, and may miss subtle regional variations. Voxel-based analysis is an unbiased whole brain approach that examines diffusion metrics voxel by voxel across the brain, enabling detailed spatial localization of microstructural changes without relying on the predefined regions of interest. It involves spatial normalization of individual diffusion images to a standard template space, and then those data are smoothed and then masked. And then there's rigorous statistical analyses performed at each voxel, and a correction for multiple comparisons is absolutely necessary to ensure validity because you're going to have as many comparisons as there are voxels in the brain. VBA is ideal for detecting global or unexpected effects in diseases, interventions, or in aging. It can identify significant group differences or correlations with behavioral measures, but it is sensitive to image registration errors, intersubject variability, and partial volume effects. Track-based spatial statistics is a widely used voxel-wise analysis method for studying white matter microstructure, and it's designed to overcome some of the limitations of traditional VBA. TBSS aligns an FA map to common template space and creates a mean FA skeleton that represents the core of the major white matter tracks. So the analysis is focused on the regions with the highest alignment, and it reduces partial volume effects. Statistical analyses are performed voxel-wise only along the skeleton, and this allows for more robust group-wise comparisons of diffusion metrics and minimizes sensitivity to misalignment and registration errors. It's particularly effective as an approach for detecting microstructural changes in white matter in conditions like neurodegeneration and aging, but its reliance on skeletonization includes or excludes peripheral tracks and may miss abnormalities that are not well captured by FA in and of itself. Pixel-based analysis is an advanced diffusion technique that analyzes white matter microstructure and connectivity at the level of pixels, which represent individual fiber populations within the voxel. FBA resolves the challenges of crossing fibers and complex geometries that traditional voxel-based or track-based methods can't. It does this by segmenting pixels from fiber ODFs or FODFs, which are estimated from constrained spherical deconvolution. FBA produces its own metrics, which include apparent fiber density or FD, fiber bundle cross-section, and the combination of these two metrics. And these offer insights into microstructural and macrostructural properties of white matter. FBA is particularly well suited for conditions with localized white matter changes because it provides greater fiber specificity and spatial detail compared to traditional methods like TBSS, for example. So now we'll go on to the region and tract-based approaches. So region-based analysis or ROI analysis is a hypothesis-driven method where diffusion metrics are extracted from predefined regions of interest or ROIs. And these can be drawn manually or based on anatomical or functional atlases. Metrics are summarized for each region, and this allows for focused analysis of specific brain regions. It's often used for hypothesis-driven studies targeting known areas of interest like the corpus callosum. Its simplicity and interpretability are major strengths as it avoids issues like multiple comparisons. However, its accuracy depends entirely on the accurate delineation of ROIs. So expertise in white matter anatomy is necessary. Plus, the ROI drawing can be very time consuming, especially when done by hand and if done on a large sample. Tractography refers to the reconstruction of white matter pathways in 3D using deterministic or probabilistic algorithms. Deterministic tractography defines a tract by following the principal diffusion direction at each voxel, whereas probabilistic tractography generates multiple potential pathways by sampling the distribution of possible fiber orientations at each voxel. Individual tracks are generated according to the manual or atlas-based definition of regions that the pathway is known to pass through, a process that is kind of illustrated down here in this bottom figure. See, there's a region down here, here, and here, and you can see them over here as they follow the trajectory of the tract. Tractography is a powerful technique for visualizing and mapping white matter pathways, offering detailed structural connectivity insights. It can be used for informing pre-surgical planning. It allows us to do exploratory and hypothesis-driven analyses. Unlike other methods, it is spatially resolved. It provides subject-specific information about fiber tracks, and it allows extraction of quantitative metrics that are averaged across or sampled along the pathways. However, its performance is heavily dependent on the algorithm used, and it often struggles with complex fiber geometries, and it is very sensitive to data quality and preprocessing. A long tract analysis focuses on evaluating diffusion metrics along the length of the reconstructed tracts, enabling spatially specific assessment of structural changes. Metrics are sampled at regular intervals along the tract and plotted in a graph as a tract profile. And this can reveal changes that may vary along its trajectory. So down here, we see the tract profiles and how FA might change along the tract itself. A long tract analysis is particularly useful for studying focal damage, like we see in traumatic brain injury, and it provides high spatial specificity and granularity, but it does depend on reliable tractography. And it can be time intensive, especially when analyzing multiple tracks. Morphometry, when it's track specific, combines volumetric and shape analysis to study the structural properties of specific white matter tracks, quantifying changes in tract volume, surface geometry, and shape deformation. It's particularly useful for investigating localized alterations in white matter pathways that occur over time, like we see in neurodegenerative diseases or developmental disorders. And it is also useful for detecting localized morphological changes in specific brain regions that may not be captured with whole brain volumetric methods like voxel-based morphometry. Additionally, it allows for precise localization of structural abnormalities along a tract, enhancing interpretations in clinical and research contexts, and linking white matter structural alterations to functional and clinical outcomes. However, just like a long tract analysis, it's dependent on accurate tract segmentation, so on the tractography. So there's some advanced whole brain approaches. The first is structural connectivity analysis, and this examines the physical connections between brain regions through white matter pathways to map and quantify the brain's network architecture. This kind of analysis involves combining tractography with gray matter parcellation, like that which is done with FreeSurfer, for example. And the resulting structural connectivity of the brain is defined by nodes, which correspond to the cortical and subcortical brain regions, and edges, which represent the strength or density of white matter connections between them. We often represent our results by a connectivity matrix, like you can see here. And in this connectivity matrix, nodes are reflected in each row and column, and edges are reflected by the value or color of each cell. Chord diagrams, like we see here, can also be used to demonstrate connectivity, and the nodes span the circumference of the circle, and the edges are indicated by the width of the chord that connect them. Structural connectivity allows us to further study network topology using graph theory. And with this, we can characterize how brain regions are integrated and segregated. Structural connectivity analysis is invaluable for exploring how connectivity patterns are altered in various pathological conditions or how they change over time. But even though it provides a detailed view of the brain's wiring, it is limited by the quality of the underlying tractography and of the cortical parcellation. So there can be difficulty in quantifying connectivity strength accurately as well. Regardless, structural connectivity is a cornerstone of connectomics, and it provides critical insights into the organization and function of the brain structural network. Connectometry is a data-driven technique that's designed to investigate whole brain connectivity in a more localized way by assessing how diffusion properties along white matter pathways are related to demographic or clinical data. As opposed to structural connectivity, which maps the global connectome architecture, connectometry focuses on the local connectome, which reflects the connectivity between adjacent voxels along a white matter pathway. This isolates localized changes in white matter integrity and makes it ideal for detecting subtle brain-wide alterations without requiring the predefined ROIs. Connectometry is used to perform correlational tractography, which identifies specific segments of tracts with diffusion properties that are positively or negatively correlated with a study variable of interest. It can also be used to perform differential tractography, which compares repeated scans and tracks segments of the white matter in which changes in diffusion metrics over time have occurred, highlighting localized regions impacted by neuronal injury or recovery. Connectometry is very versatile and has enhanced sensitivity compared to conventional tractography, but it does require data reconstructed with GQI, and it relies on accurate streamline reconstruction. It's also pretty computationally heavy. Regardless, it remains invaluable for exploring group differences, clinical pathologies, treatment outcomes, and subclinical changes in relation to white matter integrity. Here's a list of some of the most commonly used tools to analyze diffusion imaging data. All of these are free to use, and the URLs for where you can access each are in the last column. Feel free to, again, take a screenshot of this, and you can refer to it in the future when you want to use any of these. The two columns on the right that are in orange indicate what kind of interface is available to use for each program. GUI or GUI stands for Graphical User Interface, and this just means that the software has its own visualizable window that you can work in and navigate with your mouse. CLI stands for Command Line Interface, and so this requires you to write scripts or enter commands into the command line of the computer. So some level of command line proficiency is required for that software that only have CLIs like DIPI, and DIPI specifically requires Python, for example. As you can see, some programs are able to handle data reconstructed in various ways, and others are restricted to analyzing just TTI. MR-Tricks and DIPI are the most versatile tools, as they can be used with any kind of data, but they only offer a CLI for the actual analytical steps. MR-Tricks does have a GUI, but it's only used for visualization of the tracks after the analysis has been performed on the command line. My personal favorite of these is DSI Studio, which is capable of performing the most robust types of analyses on almost any kind of dMRI data. And it has a very easy and intuitive GUI in which all analyses are offered can be performed. And it also offers a CLI that is really relatively easy to learn and to use. Another great advantage of it is that it's the only software where you can perform connectometry currently, which is great. And that is all that I have for you. Thank you so much for watching, and I really hope that you learned some cool things about diffusion today.